Good evening, sir.
Hello.
Good evening, sir.
Good evening, Ajay. Good evening. Encounter problem. Excuse me, Ajay. Can you see the slide?
Sir, no, sir.
Good evening, Ajay.
Good evening, sir.
Can you see the slide? Will you please make me as a host, please? Yeah, thank you, my dear. Okay. So, good evening, my dear students. Today, what we are going to discuss about the person, a molecular basis of inheritance. There is a important portion that is called as Human Genome Project, simply called as HGP. What is that? HGP. So, what is the main use of the Human Genome Project? Why we are under the research in this part? See here, The Human Genome Project, library of life periodic table of life holy grails of human genetics. First of all, you must know the introductions. So what is meant by Human Genome Project? Where it is established or where it started? What is the main principle of HGP? And other things we are going to discuss. Okay, each and every year the Human Genome Project is also what is progressively it's going on under the research. Okay, see, the first of all, you should know what is by HGP. The Human Genome Project, simply called as HGP, which started in the year 1990. Don't forget, my dear student, this is very, very important. The HGP is started in the year in 1990 only. It's an international effort having the international effort. It means worldwide. Okay, having the principle of goal of sequencing the entire human genome. Note it down. This is very, very important. What is the main principle of this Human Genome Project goal? You know, each and every human being saw having the own specific genome, isn't it? But they want to know any one of the human beings, the particularly, okay, human beings, the entire human genomes. What is that? Entire human genomes, what it has the specification. And after that, what happened? The comparison between the nearest neighbor, okay, another human beings. And after you analyze all the human genome present in the entire human genome in the sequences of the DNA molecules, we can refer or compare with the another things. Take an example, human being having the 46 chromosomes. Okay, what are the genomes present in it? We do not know. Okay, what about the chimpanzee also having 48? Yes. What will be the similarity between these two things, and what are the differences between these two animals? Because both are mammals, right? The human beings originated from the monkey. So already you know a little about that things also. Okay, so this is the very, very important. So first of all, you should know what is meant by Human Genome Project. When, where, which it started. What is the principle of HGP? That is very, very entire human genome where we have to isolate from the nuclease, and also what is encoded, what is the amino acids, what is the polypeptide chain present in it. That things we have to know. Is it known? That is the things. Okay. Okay. What are the next one? See, this is the traditionals. Okay. When the Human Genome Project is started, now which year we are having, what are the development under the Human Genome Project? What are the development occurs each and every years? Okay, they have given us a small slide like structures. First, you should note it down. The human genome started from was where or initiated in the year 1990. After the interval of these two, that 18 years after that, what happened? You know, the formation of the Solaxa later occurred by the Illumina and the early form of NGS. This is the another point. After that, then 2002, the international, what is that? HAP map project begins. Okay, and 2003, the Human Genome Project is completed, identified. So this one is in 1990 started. When they will finish it, no, the completed ENCODE project initiated. ENCODE project initiated. And then 2005, the first GWAS completed. What is the meaning of GW? Genome. Okay, we can refer to the Google only. So I only collected the information and years only. So 2006, UK Biobank or Biobank, sorry, Biobank founded currently that 5,000. Okay, sorry, 5 lakhs participants. Okay, UK Biobank found that currently 5 lakhs participants. Okay. 2007, the first large scale of multi-disease GWAS with 5,000 participants per disease, Easton et al. Publishing first BC GWAS. This is the another one. So these are all, or what is that, each and every human beings also under the research. Those who are having diseases, in which year it becomes. Okay, what is the reason why the color of the eyes is also blue or black or brown? Why the person suffering from the hemophilia, alkaptonuria, and also other things also. Okay, so these are all are responsible for the genetic code only. That is the reason. So 2008, 100 genome project begins. 2009, schizophrenia, cholesterum, published her first PRS algorithms. 2016, Chatterjee et al. Published a PRS review. Kara et al. 2018, publishing multiple validated and PRS algorithms. So 2019, what is that? Madwad et al. Published well-powered breast cancer PRS algorithm. And the 2020, Levy et al. Published the largest GWAS to date with almost 2 lakhs participants. See that here the 5 lakhs, here the 2 lakhs participants. Recently, they were discovered. Just imagine, take an examples of COVID or HIV. That RNA containing there is a genetic code, isn't it? What are the genes present in it? That things is also isolated and also analyzed with the help of the PCR, polymerase chain reactions, and also polyacrylamide gel electrophoresis. Okay, otherwise called as electrophoresis. These are the methods that were used for the identification of the nucleotide from the DNA molecule of the different animals or plants or whatever it may be. Okay, you should know here. So this is the project starter. What is the main goal? The entire human genome we have to identify. That is the main goal of the project. Okay, so these are the years they were conducted. So many research work and also they were published. Okay, so go to the next one. Is see here. The complete in the 2003, the HGB was the 13 years project. See, started from where, 1990 started. When they will finish it, 2003, the Human Genome Project is completed. So there is given here, okay, project coordinated by the US Department of Energy and the National Institute of Health during the earlier years, the Wellcome Trust UK. Became a major partner. Additional contribution came from the Japan, France, Germany, and China, and other countries also. So all the countries also were very eagerly waiting for this project. What it has, exactly what it has, the human genome content. What is that? Okay, so that is the process. This is the institute. Which institute? The US Department of Energy and National Institute of the Health during. And also addition with the contribution came from where Japan, France, Germany, and China, and other countries also supported for this project also. When 2003 completely finished. Okay. See, what is the main project goal? This is very, very important. Okay. Identify all the approximately 20,000 to 25,000 gene in human genome. Suppose we are having 1 lakh gene, we have to identify it. Which gene, the first number, the first gene, what is that? Second gene, what is that? Likewise, 20,000 to 25,000 gene in human DNA were identified. You should identify. That is the first point, first goal. Second one, determine the sequence of the 3 billion base from the gene. 3 billion base pair that make up human DNA. Okay, that things, what are the 3 billion base pair? Okay, that things also identified. Store this information in database. After the collection of information from the previous 2 points, they may be stored in the form of database in computer. Then only the further generation, okay, in future generation, it will be used. Store the information in database. Then after that, storing of information, it is sufficient? No. They need some improvements of the tools for the data analysis. You know about once upon a time we were used telephone. Okay. After that, page. After that, cell phone. Nowadays, what happened? 4G, 3G, 5G. Okay. Even it may be so many things is also. The fraction of second, the information will be passed on to the one country to another country. That is the improvement tools. Likewise, the project related one, the human genome project related, what are the tools? What are the improved tools for the data? Improve the tools for the data analysis. Okay, it is also one of the another point that is also called as, what is that? Nowadays, you know about that. Which one you know? The artificial intelligence. Okay, the artificial intelligence also used for the, what is that? Human genome analysis. Transfer related technology to the private sector. Transfer related technology. Suppose if it is a some of the private sectors also, what happened? Technologically well versed. Okay, they can transfer the related technology and also used for the private sector also. Okay, so and then addresses the ethical, legal, and social issues. It is also very, very important. ELSI. It is mostly asked in the explanatory aspects. ELSI that may arise from the project. So these are the main goal for the, which you know, it's GP. So what are they? First one is approximately 20,000 to 25,000 human genome you have to identify. From that one, 3 billion base pairs makeup and human DNA, determine the sequence. And after that determinations, what happened, you know, identification, what we have to do now, store the information in database in the computer. And then after that, we have to improve the tools for the analysis of the data. The analysis of the identification of the genome or base pairs. Then the transfer related technologies, suppose some of the technology, take an example at forensic, okay, any other DNA fingerprintings, okay, so that sectors present in it, we can transfer the technology to the private sector also. They were used for the identification of the culprit or any other malpractice done by the person. Okay, so then addresses to the ethical, legal, and social issues. Ethical, legal. What is the meaning by ethical? You know, but there are so many things. Some person do the murders. Okay, so it will also identified with the help of the DNA fingerprinting. Okay, so this is the another things. What the next one is? What genome have been sequenced completely? It is a very, very important. What genome have been sequenced completely? Some genome that have been sequenced completely include Bacchus or Brewer's yeast. Suppose yeast also having the gene, isn't it? What the yeast it has? What the genome it has? The name is called as Saccharomyces cerevisiae. Okay, this is a fungi. A roundworm, see that? Coeno habitatis elegans. Okay. And then already you know what the fruit fly, it is Drosophila melanogastrus. Pufferfish, Fugu rubribs. A weed, Arabidopsis thalania. Don't forget, my dear, this one is very, very important. So these are the different types of, okay, different types of gene sequence it has, because each one it has a specific animals and plants and fungi. Okay, it has a specific genome, what they are having. Okay, that things they were analyzed and comparative studies also. See, the next one is the human genome important point. What is that? You know, suppose if you want to analyze the particular base pair, how much you have to spend, you know, $3 per base pair. So how many Amount, total cost, $9 billions. Okay, so 9 billions. Sometimes they may ask the question each and every base pair how many cost they were used in human genome project. Like that, they may ask the question $3. How much it will take for total cost like that? $9. $9 billion. $9 billions. Okay. So what is the method that were used for the human genome project? See that one. The method used radiating hybridization, otherwise called as radiation hybrid mapping. FISH. Don't forget, this is the FISH. Another one is called as SDSD. SDSD. ETS, pulse filled gel electrophoresis, otherwise called as, what is that? Pulse filled gel electrophoresis. And another one is called as chromosome walking, etc. So these are the methods. Sometimes they may ask the question, the human genome were analyzed, which one of the following method, like that they may ask the question. Sometimes they may ask the question, which one is the add one, like that. Okay. So this is very, very important, my dear students. Don't forget. Okay. So go to the next one. See the entire human genome have been reported in the chart aspects. First, you should note it down. The first one, human genome. Yes, human genome is nothing but a DNA molecule, right? They may be divided into two types. Already we discussed in the previous sections. One is human genome is also divided into the nature and position and also presence of the organisms, already discussed. One is nuclear genome, another one is called as mitochondrial genome. Okay, because mitochondria also it has a specific gene or DNA molecule, and also which one, bacteria also. Okay, a nuclear. What they have given inside the cell only they analyze this. This is considered as a nuclear genome, means nucleus, within the nucleus. Mitochondria means outside the cell. Okay, outside the nucleus. The human genome is the divided nuclear genome and mitochondrial genome. The nuclear genome consists of 3,300 MB megabase, about 22,000 genes here. Okay. Here, the mitochondrial genomes consist of 16.6 kilobase kp, 37 genes only. 37. See here, 3,300 MP, so 22,000 genes. And here, this one. So here, the nuclear genome is also further divided to how they will 25% of the genes and related sequences, and 75% other DNA. From this gene and related sequences, they may be divided into 10%, 90%. The 10% only the coding DNA molecule. Coding means what is that? They may be mentioned as any protein molecule or any other amino acid sequences. Okay. Remaining 90%, what is that? Non-coding DNA molecule. Non-coding do not represent any protein molecule or any amino acids and enzymes or hormone, whatever it may be. This is very, very important, my dear students. This is very, very important. How many percentage of coding DNA molecule present in the nuclear genomes? So 10%. Okay, you have to calculate from this aspect. 22,000 is the, you have to split into 25% and 75%. Divided by 25%, you will get the 25% how much. And remaining, what is that? 75%. From the 25%, you have to split the 10% and 90% also. Okay. So which one is coding? Coding how much and non-coding how much? You have to identify. Okay, this is the one part. This is also asked in the mathematical aspects. And second part, 75%, 70%, 22,000 is 100%. The 75% how much? You have to calculate it. This comes under the headings other DNA. The other DNA also divided into two top copies. See that? Single or low copy number. Single means only one gene is present in it. And middle to the highly repetitive. Repetitive means again and again. AT, AUG, like that. So there is a repetitive. How many percentage? 40% of the repetitive and middle to highly repetitive nuclear genomes present in it. Single genomes only, A only, or U only, or T only, like that. And low copy numbers. This is 60% present in it. This is 40%. Okay, you have to calculate this. This is 100%. You have to calculate 75%. From this 75%, you have to split 60 and 40. The 60% how much you have to calculate. Then again, the 40% also divided into two, tandemly repeated. This is very, very important, and dispersed repeats. Okay, tandemly repeated. What does that mean? Again and again. Okay, repeated. 100 gene present in 85 times repeated means tandemly repeated. Okay, such process is called as tandemly repeated. Dispersed repeated. Dispersed means sometimes the remaining 50% will be appear, sometimes it may be disappear. Dispersed means it may be degraded. So these are all comes under the heading is known as nuclear genomes. And next one is mitochondrial genome. It contains 16.6 kp and it's also having the 37 genes. Here divided into already 3. rRNA, 2 gene is present. tRNA, 22 gene is present. And then last one is protein coding genes. How many genes are present? 13 genes are present. Okay, 13 genes are present. This is the entire human genome project. Is that human genome is divided into nuclear genome, the mitochondrial genome. Nuclear genome contains how much is 22,000 genes. They divide into two, 25 into 75. This is 100%. You have to split into 25% how much and 75% how much. From this 25%, you have to split the gene and related sequences. That is 10 to 90%. The 10% coding gene is there. The non-coding 90%. Which one you know? From the 25%. And 75% other DNA also divided into two. That is single copy. Another one is called as highly repeated copy. 60 and 40 from the 75%. And then the middle highly repeated also divided into two. One is called as randomly repeated. Another one is called as dispersed repeated. This is also very, very important. Particularly the randomly repeated is very, very important. When you are going to study these things, we'll see. See here, the human genomes again. So the mitochondrial genome is that it is also 37 gene is that just add it all those things. 2 plus 22, 24 plus 13, you will get 37 genes. Okay. You have to add these all the category, you will get 22,000 genes. Okay. Don't forget, my dear students. This is very, very important. 2, 22, so 24 plus 13, you will get 37 genes. Okay. Okay. Got it. Any doubt in this part, my dear? Ajay?
No, sir.
So what about the Dhanalakshmi?
No, sir.
Okay. Please follow it. Tomorrow I will ask the questions. The human genome, 100% 22,000. You have to split into 25%. You have to find out, and also 75 also. From this 25%, you have to find out the 10% of coding DNA and 90% of non-coding DNA. Okay. Likewise, you have to identify it. Just note it down, my dear student. This part only. I ask you these 4 questions only. Otherwise, I will explain. No need to worry. But you practice it yourself because it is also one of the calculating method and also the mathematical question they were asked. Okay. So 100%, 22,000 you put. So 25%, 75%, how much you have to calculate. From the 25%, you have to calculate 10% is how much and 90% coding DNA and non-coding DNA. 75% you have to split the 60 and 40. So that's all. These 4 questions you must give the answer. Okay. So tomorrow you have homework. Okay. So next one is, what are the characters of human genome? After that identifications, after the 13 years, they find out what are the human genomes contain. Okay, what are the similarities? What are the dissimilarities present in it? Is very, very important, my dear. There is no without this part, there is no question in NEET. Okay, the characters of human genome. See here, the human genome contain. See here, how many million chemical nucleotide bases present in it? How much is that? So 3164.7 million chemical nucleotides. Note it down and take a notes, my dear. 3164.7 million chemical nucleotide bases. From this, the average gene consists of 3000 bases. Average means you can take any one of the gene. One gene contains 3000 base pair. One nucleotide contains 3000 base pair. Likewise, 3164.7 million chemical nucleotide base pair present in it in the entire human genome. Okay, and again, third point is the almost all 99.9 nucleotide base are exactly same in all people. Taken examples of Ajay and also Dhanalakshmi, and also taken your family members. How many percentage? 99.9% of nucleotide bases are exactly same in all the peoples, but the only 1% of the nucleotide will be very rare compared to your neighbor, your friend or family, whatever it may be, whoever may be. 99.9%, 0.1% will be differ. Okay, others same in all peoples. Okay, this is the main one. From this one, the number of protein is, how much is that? 80,000 to 1 lakh. How many number of protein present in it in your body? 80,000 to 1 lakh proteins also present in it. The total number of gene is 26.383. The total number of genes. So this is very, very important. This is also how many percentage will be similar, how many percentage will be dissimilar in nature, and how many number of protein present in your, that means what is that? This entire genome encoded these number of protein molecules. This is the point. The total number of gene is present in 26.383. Dalton, like that they told. Okay. Next, around 97% of the genome has no known function. This is very, very important. So remaining how many percentage is there? Then 3% of genome only known functions. So remaining how many percentage is unknown? 97%. From this one, which one? From this part. So the total genome from this part only how many percentage? 97% of genome has no known function. So remaining 3% only they identified. This is very, very important. See that one? Between two humans, take an example, both of you or both of other persons. Beings, DNA differ only 0.2%. Note it down, my dear student. This is very, very important. Okay. So this is the 97% of genome is unknown, right? 3% is only known one. Okay, you have to calculate that one. Comparison between the nearest persons. Take an example of two human beings. Okay. The DNA differ only 0.2% only. This is very, very important. Okay. And then next, comparison between the chimpanzee as well as the human beings. Around 98% of the DNA is similar to that of the chimpanzees. How many percentage will be differ? 0.2%. Take an example. This is also a comparison between two human beings. But in this category, comparison between human as well as the chimpanzee, how many percentage is similar? 98% is similar. So both are same. So 2% will be differ compared to the chimpanzee as well as the human being. And the next, the only about 25% of the DNA is transcribed into RNA and less than 2 encoded protein. So this is the another point. How many percentage? 25% of the DNA only transcribed. So remaining 75% is non-transcript into RNA and less than 2 encoded protein only. Then the 200 genes are close to bacteria. Okay. So remaining the human genomes, how many genomes is similar to that of the bacteria? Means 200 genes is also similar or close to the bacteria. So We are all originated from the bacteria, it's indicating. Okay. Once again, repeat for my dear student. This is very, very important, my dear. Don't forget. Once again, I repeat for you. Take a notes, please, please. Character of human genomes. The human genome contains 3164.7 million chemical nucleotide bases. First one. The average genome consists of 300 bases, 3000 bases. Almost 99.9 nucleotide base are exactly same in all the peoples. 880,000 to 1 lakh number of protein encoded. The total number of gene is 26.383 Dalton. Around 97% of genome has known function. No known function. 3% only will be known function. This is unknown function, isn't it? Between two human beings, DNA differ only the 2%, 0.2%. Around 98% of the DNA is similar to that of the chimpanzee. Only about 25% of the DNA is transcribed into RNA and less than 2 encoded proteins. 200 genes are close to bacteria. These are all the characteristics of the human genome. Not only that, and also this is the structure. You already know a little about. The DNA contains only 2 meters only, but it contains how many number of base pair, how many number of protein molecules, how it will be similar to the nearest person, how it will be similar to the bacteria, how many genes of bacteria. 200 genes. Okay, similar. See the next one. The largest known human genes is dystrophin. Don't forget, my dear. This is very, very important. The largest known human. Take a note, sir. The largest known human gene is dystrophin at 2.4 million bases. Sometimes they may ask the question, which one is the largest human genome? Dystrophin, right? Sometimes the dystrophin contains how many million bases? Like that, they may ask the question. From this one, the about 10% of genome is at telomere and around centromere. This is very, very important. Dystrophin contain how many percentage of telomere? 10% around the centromere. And then the human genome is also contain introns, or introns means in between there is a sequence is there. Introns and extrons already we studied. 24% is there. 24%. And then chromosome 1. You know about the chromosome 1. What is that? How many pair of chromosome we are having, my dear? How many pair of chromosome we are having? Ajay?
Sir.
Yeah.
Sir. So 46 chromosomes, 23 pairs.
Are you sure?
Sir, 23, sir.
Okay. You know about the 23 pair, isn't it? Pair. Isn't it? 23 pairs. The structure will be like this. So take an example. This is the first one. Sometimes they may be mentioned like this. They mention, see, like this. Okay. See, the structure will be like this. So another one is also like this. This is the number 1. This is the number 2. Like us, you have to put up to 23. The last one, what will happen, you know? The last one is called as, what is that? X and Y. Otherwise called a sex chromosome, isn't it? Sex chromosome. Last pair. The 23rd one is a sex chromosome. Okay. So this is the things. But here, how many percentage? 2.3 billion base pair is mostly present in the first chromosome only. This is the chromosome number 1. This is the chromosome number 1. This is 2. Likewise, you have to calculate up to 23 pairs. So you will get 46, isn't it? So you find out this one. So once again, I clear all drawings. So you come to the next one. See here, started from the first chromosome, second and third, and fourth likewise up to 22 pairs. 22 pairs means what is that, 44, isn't it? 44.
44, sir.
Yeah, good. 44 plus sex chromosomes. Sex chromosomes. Already you know very well, but the sex chromosome is also indicated whether it may be a, what is that? Suppose if it is, I draw here as rough calculately. Okay, rough diagram only. Yeah. So this is considered as a this is considered as a X. Okay. Suppose Y means you know about that. This is the Y. Consider as a Y. Okay. So this is the 23rd chromosomes. 23rd chromosome. Okay. What they are telling, the first chromosome only maximum present in it. That is the things they have told. In the form of which one, telomere storm. Telomere type means the structure. Telomere. Okay, telomere structures. Okay, can you understand, my dear? Do you understand? So different types of chromosome present in it. What are the types? 4 types is there. According to their structure, 4 types of chromosome is there, isn't it? What is that? Metacentric, submetacentric, telocentric, acrocentric, likewise, isn't it? So, for example, take an example. This is a chromosome. Take an examples. There is a straight one. Okay, and then second chromosome look like this one. So, short arm. This one is called a submeta, and third one is called as what is known V-shaped. Okay, these are the structures. The last one is called as what is that? Telomeres. Acrocentric means what is that? Acrocentric means what is that? See that one? So acrocentric means the absence of centromere. Absence of centromeres, how it will be? So look like this one. So look like this one. See that one? Structure will be the centromere is completely what happened now? Absence. So these are the 4 types. This is the 1 and 2. This is the third one. This is the 4th one. This is the 5th one. Okay. So these are the type, particularly the human genome. The first gene is what is that? Telomeric aspects, telomeric nature. Okay, that is very, very important, my students. See that one. The chromosome 1. See that one? The chromosome 1 has the most gene 2968. Don't forget, my dear. Chromosome 1, it contains how many is the chromosome? 2968. Note it down, my dear. And the Y chromosome has a fewest 231. 231. Take a notes, my dear. So once again, which one is the largest known human genome? Dystrophin. How many millions of base pair present in it? 2.4 million base pair. And then from this one, how many percentage of genomes in telomeric condition means 10% telomeres and around the centromere. How much percentage of intron present in the human genome means 24%. Here 2.4, here 24%. Chromosome 1, according to the structure, the chromosome 1 has the most genes, 2968 nucleated base pairs. And the Y chromosome is as fewest and 231. You must note it down. X chromosome also. X chromosome contains how many nucleated genes also present in it? Just note it down, my dear student. This is also another question. Next, see that one? The repeated sequence from that one, repeated sequence, they do not code for protein. The repeated sequence is also there. The genes are called as junk DNA. Junk DNA is also called as repeated sequence. A repeated sequence is also called as junk DNA, but they will not code for any protein molecules. Okay. The junk DNA, how much is that? See that make up at least 50% of the human genome is junk DNA. That is denoted as the repeated sequence. Okay. So this is very, very important. It's slightly confusing, but at the same time, don't forget, my dear. Repeated means, I already told you. Take an examples of first codon, first number. Okay, AUG. And take second, fifth one is AUG, 10th one is AUG, and also likewise repeated again and again. How many percentage means 50% of the human genome is repeated, also called as junk DNA. Okay, next one. The scientists have identified about 1.4 billion location where a single base pair. From this point, from the DNA molecules, right? Scientists have identified, right? How much? 1.4 million locations. Location means, but each and every gene present in the a specific locus, a position, isn't it? That is called as locus. That location where single base pair only. Single base DNA differences occur in humans. This is very, very important. So this is also I show you, so we can identify that things also better understanding. See, take an example. So this is the DNA sequences from this one. Okay, from this one. See that. Okay, from this one, this is the entire nucleotide. Take an example, 2 meter like this. Here they mention as a triplet codon, isn't it? So likewise, you can imagine. Likewise, isn't it? So this is the one, this is the fifth one, this is the 10th one, and 15, 20, like that. Imagine. Here they encoded triplet codon. Triplet means AUG. Suppose if it is the nucleotide present in the AUG means this is the one locus. This part is called as one locus. Number 1, 2, 3, 4, like that, isn't it? This is mentioned as a methionine, isn't it? Methionine. Met denoted as methionine. What they are telling now, from this one, 50%, what happened, single base pair. What is the meaning of single base pair? Here the triples, isn't it? Triple. 3 codon present. Single means, what does it means? Any one only. See that one? Any one. Any one means, take an example, 5th one is there. Here the 8th one, it's considered as only A only present. A. Okay. Here the 12 is also A, likewise. Here also T. So likewise, you can calculate it. So different according to the locations or loci. Okay, how many percentage? 50% of the genes are, what is the genes are? How it will be? Single base. Single means so only one base pair is there. Single. See that one? This is a single. Not only adenine. So just I mentioned here adenine only, but not only this. Sometimes it may be present in the some other components also. Some other means, take an examples. Here you have take it. So the third one is a, what is that? A guanine. Okay. Here the 9th one is guanine. Okay. So here the 12th one is guanine. So these are all a single base pair. Okay. So take an example. This is single. So G. Okay. And also A. So these are all single. Okay. So suppose if it is a triplet means, see that this is triplet. So this is triplet. See that here triplet A U G. See the next one here, that C C C. So whatever it may be, like that. Okay. So it located, this is single base pair located in between the triplet codon. That is the main important. This is also 50% of the codon is the triplet codon, and remaining 50 is the single codon in the entire sequence of there. That is the point. Okay, don't forget, my dear students. This is very, very important. Okay, see. See here? See that. So that gene is called as junk genes. Okay. Next, the scientists have identified the 1.4 million base located in the single base, but 1.4. This is very, very important. Okay. So this is 50. Sorry, extremely sorry. The 50% is called as what is that? Junk DNA, otherwise called as non-coded protein molecules. The 1.4 million base pair is, what is that? Single nucleotide base pairs. Okay. So this is very, very important, my dear. So next we will pass on to the next one is bioinformatics. This is very, very important, my dear students. Nowadays, the research is also going on. Bioinformatics is very, very important. What is by bioinformatics? What already you have to identify in the different types of animals or plants. You have to store whether it may be a protein sequence or other DNA sequences or hormones, enzymes or vitamins or whatever it may be. Vitamins it not comes under this getting the sequence aspects. Adenine, guanine, thymine, cytosine, whatever it may be, bioinformatics. See that one? The bioinformatics is an emerging field, consists of molecular biology. What is the meaning of molecules? What is the meaning of biomolecules, my dear students? What are the biomolecules present in our body? Ajay?
So proteins,
biomolecules.
vitamins,
Vitamins, yeah.
So
Protein.
carbohydrates.
Very good. So that is the thing. The lipids also very good. Carbohydrate, protein, lipids, and vitamins and minerals and the hormones, enzymes, like that, isn't it? These are all biomolecules, but here what they are, molecular biology. Molecular biology, what are the molecules present in your body? Okay, that molecules are molecules containing what are the component? Carbon, hydrogen, basically carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. These are the basic things. And the bond will be already you know about what is there? Hydrogen bond is there, sulfur bond is there, and also glycosidic bond is there, peptide bond is there. These are the bonds. Okay, that information, particularly the bacteria contain how much is that 200 gene is similar to that of the human being. How they are identified, how they analyze means with the help of bioinformatics. Comparisons. See that one? And also store in the form of database in computer. That is bioinformatics. What is that? Human genome is contained 2 meters. Okay, how many nucleotide present in this much? How much is encoded protein? How much is non-encoded? How much billions is single base pair? Okay, how much is similar to that bacteria? How much is intron is present in? Likewise, there is a information. That information stored in the computer that is called as bioinformatics. Simply, see that one. Bioinformatics is an emerging field, consists of molecular biology and computer science. Both are okay. That centers on the development of database, computer search algorithms, gene predictions, software, and other analytic tools that are used to make sense of DNA, the RNA, and the protein sequence date. This is very, very important. This is called as what is that? Bioinformatics. What are the protein molecules? DNA, RNA, and protein molecules also that these are the biomolecules. We are collecting the informations about the molecules. That process is called as bioinformatics. We are getting the information about the particular biomolecules. Okay, see that one? You can identify. See that? Bioinformatics. It has several branches in there and also divided into so many branches. It's also included computer science. Without computer science, we cannot be able to identify the sequences. Storage ability is not possible. Chemistry. Chemistry is the part of the bioinformatics because chemistry is very, very essential because each and every living organism are made up of chemical substances, whether living or non-living, whatever it may be. Biochemistry. Biochemistry means, what is the difference between chemistry and biochemistry? Chemistry means, what is by biochemistry?
So normal study of chemicals is chemistry, sir.
Okay.
Biochemistry is when we study compounds with respect to our body, sir.
Yes, the chemical reactions which occurs within the two components is called as chemistry. The chemical reaction which occurs within the living molecule is called as biochemistry, isn't it? Okay. And biology. Yeah, biology, the science that deals study of the plant as well as the animals. It is also because where we need bioinformation means biology, living organism only, whether it may be plant or animals. Then we come to the conclusion, statistical analysis. Statistics is also it is also included the bioinformatics, because then only we can identify this millions. Also, human genome is similar to that of the chimpanzee or any other plant, or like you have to calculate. We can engineer. What is the meaning of genetic engineer? Genetic engineer, gene engineer, genetic biotechnology. Okay, bioinformation. Genetic engineer means we can create our own gene sequences. How it will be? There is a special methodology is there. Cutting, crafting. Likewise, the genetic engine, gene cutting is also there with the help of the endonuclease. Restriction endonuclease is there. After the removal of the particular gene sequence in the foreign particles, and well-known and beneficial DNA molecules also inserted with the help of the ligase. And also the amplification takes place where the lower vector is called as E. coli. So finally, what happened? The entire sequences, it will be produced in the within the lower molecules. So this is all, these are all our comes under the headings known as what is genetic engineering. So our next chapter, that one, biotechnology and genetic engineering, we will study that things is also. Okay, see here. So this is the bioinformatics. The symbol diagram has given here. See that here the biomolecule is there. This is Golgi apparatus having the nucleotides, otherwise called as gene sequences is there. The gene sequences, they are having the nucleotide or whatever it may be converted into what is that? Machinery language, and also feed into the computer. This is called as bio and stored in it. Okay. When we need, we will use it. This is called as bioinformatics. Okay. So next one is very, very important, my dear. The bioinformatics also used in pharmacology. Okay. That's why it is called as pharmacogenomic. Okay. So here the spelling mistake is that pharmacogenomic. Okay. See here. So pharmacogenomics is the study of how an individual genetic inheritance affect the body's response to drug. This is very, very important, my dear. This part is very, very important. Okay. So this is very, very important, my dear students. What is that meaning? The study of how an individual, any one of the person, individual person, genetic inheritance. Genetic inheritance means the character transfer from the one generation to another generation. Then that gene is how it responsible for the drugs. Because you know about each and every person, they will not accept. Suppose, Ajay, your body accept the penicillin injection.
So,
penicillin injection, your body is acceptable or non-acceptable? Not acceptable.
So it accepts, sir.
What about Dhanalakshmi?
Yes, sir, it accepts.
But I am not. It is not accepted for me. Okay. So because there is a variation is that there is the reasons. The individual genetic inheritance also affect the body's response to drugs. Okay. The term comes from the words pharmacology and genomic, and it is thus an intersection of the pharmaceuticals and the genetics. Okay. This is the pharmacogenomic. Okay, how it will be response? See the slide, you can easily identify. This is the normal individual one. He has a DNA sequences like this. Okay, already know it. It has a own specific genome. Okay, the medicine responsibility will be only one tablet is enough for the any other disease like this. No, just imagine. Okay, some of the person, they may took only one tablet. Some person they may took half tablets. Some person may took 2 tablets. Some person they will not take any tablet, but the disease will be cured. So these are the variations. Okay, see the dosage and medicine selection based on the genomic profile. How they will be identified? The person is suffering from any one of the head or fever or whatever viral fever, they take only one tablet, it's enough for this person, and half tablet for this person, 2 tablets for this person. And sometimes without taking any tablets, will not took any tablets, what happened to the disease will be completely curable. How it will be possible? These are all are interrelated to the, which one you know? Pharmacogenomic. Okay. Suppose one person suffered by a headache, they take or they took, which one you know, one tablet. The same fever, some person they take, which one you took, which one you know, half tablets. Sometimes 3 tablets is also, it's not sufficient. Okay. So these are all, or what is that comes under the heading is known as pharmacogenesis. See that what are the benefits of pharmacogenomics? You know, more powerful medicines, more powerful medicine we can produce, or we can reduce the power. Okay, first point. Then second one is between safer drugs the first time. This is also another one. Okay, safer drugs means, you know, any one of the drugs introduced in the market, before introducing the market, first the scientists checked where? In the laboratory aspect, in vitro or in vivo. In vivo or in vitro, my dear, within the lab only, isn't it? First they check from the lower animal, take an examples of the rat or rabbit or whatever may be, then it will be entered into the chimpanzee. So these are all are considered in mammals, isn't it? After that, there is a test person is also there. They give test drugs for the particular person. And also age group is also there. Age group also, lower age, 1 to 5, and also child also, fetus. Fetus they will not provide, particularly the age group is also there. Okay, up to 5 and 10, and then teenage, and then men and women, and also pregnancy during lactation period, like that, they may be divided. They may be given and also analyzed for the long period. Then only they come to the market. Okay. And then advanced screening for the diseases. This is also another point. Better vaccinations. Already you know about. Already you put a COVID 1, 2, 3. Okay. And dosage, booster, like that. You know how it becomes, you know, due to the 100 millions of checkup. After that, after the introduction of the human beings, and also what are the drawback is that all are corrected, and finally it will enter to the market. This is very, very important, my dear students. Okay, see the next one, you can identify, see that. So the same DNA molecules, so the same drugs, 3% have taken here. So one is slow metabolized. Sometimes the drug is some person they may be slow metabolized, and this is one is a normal metabolism. Slow metabolism 25% or 20% like that. This is the 50%. This is the normal metabolism. Rapid metabolism. Suppose when you are taking the tablet, within few minutes, the fever is completely recovered. Okay, so it will take 4 to 5 days. It will take 2 days. It will take 1 day or 1 hour. So these are the differences. How they will be identified? All are under the test only. Okay, so we will finish it up to this level only. We will see the remaining things on the next classes, my dear students. DNA fingerprinting. We will see the next class. Today, what we discuss, I summarize all those things. See that first one is a human genome project in the 1990. What is the main principle of this one? You know, the entire human genome we have to identify and also analyze. When they started in 1990, and also 2003, the human genome project is completely finished after the 13 years. Don't forget. And complete genome project already know what are the supporting countries also there, how they support our contribution is there. Okay. And project goal, what is that? You know, approximately 20,000 to 25,000 genome in human beings we have to identify. Second, 3 billion base pair make up a human DNA. Store the information in the form of database, not in the human being and also other animals also. Just to go and check into the DNA library or DNA fingerprinting. DNA library will get the information. And improve tools for the data analysis. Transfer related technology to the private sector. Just to take an example of central government to the private sector means any other police department or or any other bomb squad, whatever it may be. Okay, and this is also ethical, legal, and social issues also there. Okay, and then so what are the human genome have been sequenced completely? Means these are the test samples were taken, and then cost, how much it will be? $3 and entire $9 billions. And what are the methods? This is very, very important, my dear. This is the method they were used. So this is very, very important. Okay, so radiation hybrid mapping, FI-sketch, and STDS, ST, and ETS, and pulse field gel electrophoresis, chromosome walking, etc. Okay, tomorrow you must note it down. This point is, don't forget, my dear, you have to find out these answers also. Human genome, how it will be divided, nuclear genome, mitochondrial genome. Nuclear genome is divided into two, and genes and related sequences, other DNA, so 25%, 75%, and gene sequences also divided into coding and non-coding, 10 to 90, other DNA, 60%, 40%, single lower copy, and also middle high repetitive, and tentative, and dispersive. Mitochondrial, it has a 37 gene is there, and rRNA is 2 gene. Don't forget, my dear, rRNA 2 gene, tRNA 22 gene, and protein coding in 30 genes. If you add all the three, will get the 37 genes. Okay, that thing's also very, very important. And then character of human being, already know all about. How many is millions of chemical nucleotide, and average gene is how much, and almost how many percentage of nucleotide is similar to that of the other people's also, 99.9%. How many protein molecules encoded the nucleotides? 80,000 to 1 lakh. And total number of genome is how much? Already you know all about. Around 95% of gene has no known functions. Okay, and between two human beings, how many percentage will be similar or differ? And around 98% of DNA is similar to that of the chimpanzee also. 25% of the DNA transcribed into RNA, and less than 2 encoded protein. 200 genes are close to the bacteria. This is also very, very important. And then 10% is telomere structure according to the structures around the centromere. And the largest human genome is very, very important, dystrophin at 2.4 million bases. Chromosome first has the most genes, 2968, and Y chromosome 231 chromosome is the repeated gene is called as how much is the 50%, that is called as junk genes. Another one is 1.4 million location where single base per DNA present in it. Next one is bioinformation, collection of information from the different animals or plant. They may be stored in the computer, particularly the biomolecules, DNA and RNA protein sequences. So what are the interrelations? Subject is there, and also what is the bioinformation, how it will be stored in the things, and also pharmacogenomic, how it will be used for the pharmacological aspect, how it will be used, more beneficial aspect. So more powerful medicine between the safer drugs the first time, advanced screening for the diseases, and better vaccines have been identified. So that's all about my today, my dear students. We will see the remaining things on the next classes, my dear. Thank you so much for the participation. Thanks. Thank you, ma'am. Thank you, Ajay.
Thank you, sir. Thank you, sir.
Thank you, sir.
Okay. Take care. Bye.